Hi guys, thanks for coming. I'm super excited to be your Master of Ceremonies today. I just found out like an hour ago that's what MC stood for. So this is going to be a good. Like Jess said, we're really excited to have such a great turnout. This is the first time we've tried this as a department, so we're really excited to see how it goes. I know Megan's been great about getting word out to this department and other departments, and someone called up Jess the other day and was like, hey, is plant biology having a fight next week? So we've got some good publicity. You know, there's been some debate about whether this is a throwdown or a smackdown, but I thought I'd let you guys decide for yourself, because either way, it's going to be wild. So, like Jess said, it's the 2014 Plant Biology 3-minute throwdown. Well, I guess that solves that issue. It's a throwdown. So before we get started, I wanted to introduce you guys to the rules and then the judging criteria for how this throwdown is going to work. For the presenters, you will each be allowed one slide, and that includes no animation, no sound, and no video. Also, no props, except for your hands. And most importantly, your presentation cannot exceed the 3-minute mark, and that's absolutely critical. And Carlina has graciously volunteered down in front to keep track of time. So she's got a comically large stopwatch on her iPad that'll count down from 3 minutes. But if you go over the 3-minute mark, you will be automatically disqualified. So no pressure, but it's a 3-minute thesis competition for a reason. I guess it's just hanging out in one of the student offices. I would agree with that. The presentations will be judged on 3 main categories: comprehension, engagement, and communication. And this is basically, you know, did the presentation make sense? Did the audience understand it? Was it engaging and made people want to hear more? And was it communicated in a way that was effective for a non-specialist audience? And this is a really important part of scientific communication. So these are criteria that you guys should keep in mind when you're voting for your People's Choice Awards. Did everyone get one of these neat little ballots, since you'll be doing some voting today? And if you don't have one, make sure you get one. So you will be able to help contribute to naming one of our winners today. But because plant biology rolls hard, we also decided to bring in some professional judges to make this the real deal. So I'd like to take a moment to introduce our judges. First, we've got Dr. Katrin Stanger-Hall, who co-taught a science communication seminar in the plant biology department a couple years ago that Jess and I took, and it was awesome. And she also participated in judging the 3-minute thesis preliminary rounds, the one that the grad school does. Next is Dr. Stephanie Pearl, who is a newly minted doctor from our department, so welcome back, Stephanie. She also co-taught the science communication seminar with Dr. Stanger-Hall, and she's also a co-organizer for the Athens Science Cafe that Jess talked about. And the flow of this is very nice because she is also a co-organizer of the Athens Science Cafe with our next judge, Mr. James Howdaway, who works for the OVPR, and he also helped with the 3-minute thesis judging for the grad school, and is involved with lots of science communication courses, like our 4th judge, Mr. Anthony Murata from the Department of Theater and Film Studies, and he's the one who teaches the GRSC course on science communication, which a lot of plant bio grad students have taken, and they said it's awesome. So that's basically it. That's my spiel. Does everyone understand the rules? Is everyone excited to see some rapid fire science? All right. In the spirit of a smackdown, I just have to say, throwdown. In the spirit of a throwdown, I'm going to say, let's get ready to botanically rumble! Oh, wait. I just remembered. One more thing. Please hold your applause until the ends of all the presentations. So we'll have an easier time transitioning between participants. So just hold all your applause to the end and then go nuts, okay? Question.
Are you all going to position the official clock so that the speaker can see it?
Carlina will hold them up so the speakers can see the 3-minute countdown. All right? So without further ado, I'd like to introduce our first contestant from Entering the ring from Team Grad School, we've got Miss Chelsea setting the bar high Kennard from the Lankow Lab. And she's going to talk to us today about something entitled Attack of the Invasive Plants. Can they ever be stopped?
Woohoo.
All right, Chelsea, take it away.
Thank you. When you think of invasion, the first thing that probably pops into your head is aliens from outer space. But when I think of invasions, I automatically picture plant species from this planet. Although we may think of different things, alien and plant invasions do have some things in common. First off, when alien and plant invasions both require the movement of the species into a new area from its native land. Second, when aliens and plants invade, they become the dominant species in the community and cause all sorts of problems. Invasive plants can alter ecosystem services like decreased pollution mitigation and local biodiversity. For my dissertation, I decided to study the invasive plant over the alien because they're a little less scary and easier to find. I have chosen to study an invasive grass, Japanese stilt grass, and whether it's able to maintain being an aggressive invader through time. Scientists think that one reason why certain plant species are able to become invasive is because they escape their specialized pests and pathogens. In the native range, these pests and pathogens, like insects and fungi, are able to stabilize the population. But when these plant species move into their invasive range, they escape these specialized enemies, and this allows for explosive population growth. However, it's unclear for how long this actual release from enemies can last for. It's quite possible that these enemies can catch up to these invaders, either by coming from the native range or developing in the invasive range. In my research, I will explore whether this invasive grass accumulates soil fungal pathogens through time, and whether this can be related to a decline in the grass's health. So far, what I have suspected is supported by my data. I have found an increase in mortality at older invaded sites, and this is correlated with the change in the fungal community in the soil. Although I still have some work to do to figure out whether this change in the fungal community is actually pathogen related. This research is really important because understanding how invasion plays out through time can actually help in management goals. If invaders are becoming less aggressive through time, then the problems they are causing are probably decreasing as well. So targeting the younger populations would be ideal for managers. So although I'm not saving the planet from alien invasions from outer space, by doing my research, I am aiding by a large impact on real-world plant invasions. Thank you.
Hold on now. What did I say? All right.
Can't help it.
Thank you, Chelsea. All right. See, not bad, right? Get all the anxiety out. It's a good thing that was only 3 minutes. Otherwise, it would have started growing on me. All right. Enough of that. So now we've got our first participant from Team Faculty, Dr. Chris Peterson, and he's going to shake things up a little bit with his presentation called Using Tree Wind Damage to Improve Meteorology's EF Scale.
All right, so most of you know that I've been studying windstorm damage to trees and forests for most of my career. During that time, I've realized that the meteorological community has a problem, and that is one of their most dangerous phenomenon, tornadoes, we all know about them, are very hard to study. They're short-lived, they're very unpredictable, very hard to study in real time unless you happen to be parked in the right place with your Doppler radar pointed at the tornado. It doesn't happen very often. So as a result of that, they're forced to sort of rely on retrospective approaches to study tornadoes. One of which you've probably all heard of is the F scale, or the EF scale is the new name. This is a damage scale in which tornado wind speeds and tornado behavior is inferred based on damage to buildings or structures or, in fact, trees. This is where I come in. I can help them. So the problem is the EF scale it has a number of criteria for rating tornado intensity. Most of those criteria are based on buildings. Some of them are based on trees. Unfortunately, the tree part of the EF scale is deeply flawed. It has a lot of problems, and I've been telling them this for a couple of years now, and they're starting to listen, thankfully. But instead of just sitting on the sideline and complaining, I thought, how can I help them? Because I do know a little bit about trees. And the answer seemed to be help them calibrate the tree part of the EF scale. And so how do we do that? That's what I'm going to talk about here for a minute or so. And that is to go to a post-storm situation and compare side by side damage to houses, which are understood really well from an engineering standpoint. We can be pretty confident of our estimates of wind speeds based on house damage and calibrate that to the trees that are right next to the house. So here's a couple of examples. Here's a house that has EF2 damage. Here's a tree that has hardly any damage, and so that would be rated EF0, so that's sort of a problem. So these two pictures kind of illustrate the problem. The answer is to go through for a large number of houses. This is a project that's still underway. And compare the damage to houses and the trees that are right next to them, and calibrate the tree damage such that we know what the wind speed was for the house, because that's understood very well. And then we can calibrate the types of damage to the trees, so whether the tree is intact or standing or defoliated or branches broken or whatever. So this is a way that we can improve on the EF scale for the meteorological community, hopefully save some lives in the process, and make a contribution from the ecological world to the meteorological world. That's it. Thank you.
All right.
This clock is terrifying.
If you guys are really excited about a presentation, just... Woo! All right. So next we've got someone from Team Grad School. We've got Ed Macattack Mackesy talking about another very important issue. Here you go, sir. And he's going to talk to us today about climate change, problems, predictions, and what we can do. All right.
Thank you.
Right button when you're ready.
All right. On Friday, President Obama set aside $183 million for the western part of the United States. This upper panel shows why. This is a map of drought in the western U.S., and the darker colors here represent more pronounced drought. So you can see they're really focused in California, and this has really big impacts on crops, livestock, as well as drinking water. While we can't generalize from a single weather event to global climate change, this is certainly adding to a body of evidence that these drought periods are becoming more and more common and are certainly suggestive of global warming. So what we can see up here in the upper right is a prediction. It's a computer model comparing average temperatures in the year 2100 to present day. And the fact that it's orange and red indicates that we predict it's going to be warmer. The temperatures are, on average, going to increase over time. So as plant biologists, we're concerned. We want to understand when and where we can plant our crops in this changing environment. And this is a crucial question. To get at that question, for my dissertation, I'm studying wild sunflower. It's a species with a large range, so it encounters a lot of different environments, all the way from Mexico up into Canada, actually. And so what you can see here are actually two of the extremes. This is a Texas individual and a Canadian individual. And the first thing that jumps out at you is just the huge size difference. Texas plants are taller. But there's a couple things you can't see. One is that I planted this Canadian individual 2 weeks later compared to the Texas individual, so it flowers more quickly. We think this is an adaptation to the short growing season seen in Canada. Another thing you can't see is that their fatty acid profile is very different. We think that the fatty acids in Canada derived plants are actually adaptive for their germination in cooler temperatures. So we think we can understand what an adaptive phenotype is in the wild populations, and maybe this can help with adapting crops to climate change. But wild populations are also a tool for outreach, and that's what we're seeing over here. As a part of a module I've developed for Hillsman Middle School, I'm growing wild carrot and lettuce, as well as high-yielding cultivars. And what we're going to do is keep some well-watered drought others, and let the students measure the phenotypes. Hopefully they take away two things. One, hopefully they are in awe of the domestication process. Oh wow, this is what a wild carrot looks like. But beyond that, hopefully they can see the impact of climate change on the food we eat, and hopefully that builds a lasting connection, and maybe they'll be the future scientists to tackle the problem. Thanks.
You guys are scared of me. I like this. All right, good job, Ed. All right, that was a pretty hot topic, right? But there is a purpose for that segue, because speaking of hot topics, we've got Dr. Shumei Chang here to talk to us about pollen competition. So in style. So she's going to talk to us about, is bigger always better? All right, don't laugh too loud.
Okay, so actually I'm going to switch, sort of the kingdom here a little bit and get you to start thinking about moose. Okay, so when you think about moose, what do you think first? It's ridiculously large antlers, right? So sexual selection is actually, this theory was suggested by somebody, by Darwin, to explain these kind of ridiculous and spectacular traits in animals, okay? So the idea here is that these traits are good for male-male competition, they butt heads, and for female choice. These female individuals actually basing on these traits to select which male they're going to mate with, okay? So these two processes, male-male competition, female choice, are really well accepted in animal literature. But when it comes to plants, people doubt it, okay? They said, oh, well, you look at these morning glories, they are so pretty, anything that can attract pollinators to come to this plant is going to be good for male function, good for female function, and therefore you should not see anything that will be sex-specifically operating. Okay, so I beg to differ, because I think what we are focusing right now is actually these really flashy traits that happen before the pollinators come. But what's important is actually after the pollinators have come. Okay, so these pollinators, they bring pollen grains, they come to the stigmatic surface, this is my representation of stigma, and they say, okay, we have different pollen grains coming from different individuals, now ready, set, go. See who is going to run faster, grow faster, and reach the girls first, right? And so that's what I think sexual selection can be applied to plants, okay? So what I did was that we developed these two lines of pollen grain lines. One has larger pollen grain, one has smaller pollen grains, and we put them on the stigma. And I hope you can see that these little thin lines are actually pollen tubes that are growing out of pollen grains, okay? They're racing down the style tissue to reach the female, the ovules, okay, of these plants, okay? And so the specific question that we're asking in this line of research is pollen size, which is the trait that I could find that is as male as you can find in a hermaphroditic plant, okay? Is larger pollen grains always better than small pollen grains when it comes to getting the girls, okay? And so we did this. We brought larger pollen grains, small pollen grains, put them on the stigma, and then we look at the seeds that came out from these pollen competition, male-male competition, and what we found was that indeed larger is most often better, but not always, okay? So we found 78% of the seeds that actually came from, is that what I have left? Okay, so given that result, we want to know why that happens. Thank you. That's all I have. That's horrible. I thought that's what I used, but that's actually what I left.
Thank you. Oh boy. So if it hits 0, are you just gonna like throw something up or just wave? All right. Oh, all I can say is I po me oh my Shumei. That was absolutely morning glorious, don't you guys think? All right. I kid. Now, all right, next up from Team Grad Students, we're going to heat things up a bit with our presenter, Jeff doesn't need a nickname to sound like a SmackDown champion, Cannon, from the Peterson lab. And he's going to talk to us today about crash and burn, interactions between forest wind damage and fire. There you go. And right button when you're ready.
Press what now?
Right button when you're ready.
Gotcha. So my name is Jeff Cannon, and I study how forests are affected by wind damage and fire. And so disturbances like hurricanes and tornadoes and wildfires are really destructive and cause huge loss of life and property, as we saw with the Moore, Oklahoma tornadoes recently and the Arizona fires this summer. And so because fires can be so destructive, it's really important to the public as well as to ecologists that we understand what determines how these disturbances behave. Current research suggests that the way a disturbance behaves depends in part on what disturbances have occurred previously. So you may expect a wildfire to be really severe if just before the fire a tornado came, toppled lots of trees, and created lots of additional fuel. But fuel isn't the only important factor governing fire behavior. There are other important aspects like weather and topography that can have an impact, and other fuel characteristics such as the amount of fuel, the type and size, its moisture and arrangement can be really important as well. So what my research does is look to see how tornado damage can impact these fuel characteristics so that we can better predict how wind damage will influence a subsequent fire. So in order to do this, we've worked in a forest where we've done some simulated wind damage and a controlled burn, and we've found some pretty surprising results. So first of all, the most obvious thing is we saw a huge increase in fuel after the tornado damage, and that led to a really intense fire. But what was interesting was that the increase in fire intensity occurred for reasons other than what we expected. Yes, there was a huge increase in fuel, but most of that was in the really large size classes like downed trunks and branches that never actually burned. So if you exclude those really large size classes, the available fuel is actually pretty similar in amount between damaged and undamaged areas. What changed was the makeup of fuel. So you had a small increase in the amount of fine woody material and grasses in the damaged area. So that gives some explanation to the increased fire intensity. But what was really drastically different between the damaged and the control areas was there was a huge rearrangement of fuel. So in the undamaged areas, fuels tended to be spaced out really evenly distributed, whereas in the damaged area, fuels were clustered tightly together in some areas and really sparse in others. And so if you think about a campfire, it's really easy to see how this rearrangement of the same amount of fuel can lead to an increase in fire intensity. Because you've got fuels clustered close together, the heat from one fuel element can preheat and dry out the next piece of fuel so that the entire pile of fuel can burn more completely and with higher intensity. So that just goes to show that the interaction between wind damage and fire is much more complex than a simple addition of fuel. If we want to understand how destructive disturbances like fire behave, we want to understand not just the changes in fuel amount, but the more complex mechanisms through which disturbances interact. Thank you.
Thanks, Jeff. All right, that was also pretty hot, guys. Now, okay, don't get me wrong, Jeff is great, but if you're interested in a real fun guy, look no further, because representing team faculty, we've got Dr. Michelle Momini, and she will be talking to us today about exploring the septon cytoskeleton and a fungal model system. Right?
I gotta correct you though. I'm a fun gal. So which one is it?
Right button.
So the cytoskeleton is a series of scaffolds inside of the cell, and these scaffolds support the cell, they shape it, and they help organize all of its activities. Septons are a really recent discovery that play a part in this cytoskeleton. Now it turns out that humans have septons in virtually every cell of the body. When septons are missing or damaged, there's several diseases that can result, and those are symbolized by the ribbons up in the top corner. So leukemia and lymphoma, Alzheimer's, bipolar, schizophrenia, all have been associated with damage to the septons. We don't know if damage to the septons is causing these diseases. We don't actually even know how septons are organized or what else they interact with in the cell. Trying to figure out what septons do in a human is a little bit like trying to figure out what steel does in the world's tallest building. The Burj Khalifa, pictured over in this corner, is 2,700 feet tall. It's 163 stories, and it has 55,000 tons of steel. Probably not the simplest place to figure out what steel might be doing. Enter the humble fungus. Yes, bread mold, shower curtains, toenails, that kind of fungus. In the bottom center here is Aspergillus, the fungus we work on in my lab. And fungus is to human as cottage is to the Burj Khalifa. Smaller, simpler, but they have many of the same basic systems. So in one set of experiments in my lab, what we did was take a green glowing tag and add it to a septon. And you can see that result here in the center. In the same way that a string of Christmas lights will show you the position of a roof line, this tag shows you the position of septons within the cell. And what you see in this particular example is a very young fungal cell with one tube coming out, and the septons are forming rods along the outside of that tube. In another series of experiments, we took septons and removed them from this fungal cell. We expected that the cell might collapse, but what we saw instead is pictured in this bottom corner. Instead of seeing just one tube emerging from this young cell, we now saw multiple tubes emerging. So we're working right now on trying to understand how the loss of a septon is leading to uncontrolled growth in this fungus. And we think that understanding this will help us better understand the connection between septons and uncontrolled growth in human diseases like cancer. Thank you.
Thanks, Michelle. I can really see why her research gets a lot of support. All right. Next up from Team Grad Students, we've got the guy who's answering some of the big questions. His name is Aspergillus harkesi, but you guys might know him better by his common name, Alex Harkis. And his talk is called Why the Why. Button when you're ready.
I dedicate this slide to Kelly Daw before I begin. Likely, most people in this room have 23 pairs of chromosomes. One of these pairs is called a sex chromosome. It's what genetically determines you as either a female, having 2 X chromosomes, or a male, having 1 X and 1 Y. Now, under the microscope, the X and the Y chromosome look very different in humans, the Y chromosome being about 10% the size of the X, and having only about 78 genes, relative to the X chromosome's nearly 2,000 genes. So what has been driving the loss of genes on the Y chromosome relative to the human X? Well, we know that this has been happening over the last about 200 million years. What we can't do, though, is go back in time and look at what the Y chromosome used to look like and identify mechanisms that might be initiating this massive loss of genes on the Y chromosome. What we can do, though, is, this is important to us because at the current rate of gene loss on the Y chromosome, we may be facing a possible extinction of the Y chromosome in the next 10 or 20 million years. My research is dedicated to all the men out there. What we can do, though, is look at systems that have very young sex chromosomes, and we can see what does a very young Y chromosome look like and what might be the mechanisms that drive potential degeneration of Y chromosomes relative to the X. I work with asparagus, something that you might find in the vegetable aisle of your grocery store. Asparagus is a very special plant because it's got very young, identical X and Y sex chromosomes. Just like in humans, XX makes a female plant, XY makes a male. Now, when we look at the X and the Y of asparagus under the microscope, very unlike humans, they look identical to each other. But when we look a little bit closer at the DNA level and we compare the X to the Y and look along its length for differences that might be specific to the gender, what we see is that the Y chromosome is a little bit different from the X. And that specifically, the Y chromosome has a region bound on the left side and the right side that contains genes that are essential for maleness. Now, in this region, it essentially is acting like a trap. We know that these genes need to be on the Y chromosome, so the Y will do everything to keep them in place. Now, like any good trap, things can come in, but things can't come out. So when we look at the Y chromosome relative to the X, what we find is that on the Y chromosome, these potentially harmful DNA elements called transposons are accumulating in this trap on the Y, and they're not being able to be let out. Now, what this eventually is ending up is that maybe the Y chromosome Y chromosome is going to accumulate as many of these elements as possible, which eventually may be harmful to the Y. So perhaps a more terrible story than anything is that maybe Y chromosomes are born to be destroyed and that there's no stopping this eventual degeneration of the Y chromosome. Thank you. Is that a happy way to go out?
I think we can all understand why Alex's research is so cool. It's like a history lesson on a chromosome. But if you guys are interested in history, boy, do we have a treat for you today. Dr. Andy Patterson is going to be talking to us about a brief history of plant genome evolution.
So some of my earliest childhood memories are of the fruit and vegetable gardens of my parents and grandparents. I just couldn't get enough of Pop-Pop's raspberries, of Grandma's applesauce, or of Mom's tomatoes. Moreover, even as a child, I was fascinated by reading seed catalogs, looking at pages and pages of different cultivars of tomato and cucumber and pepper in different sizes and shapes and colors and properties. This interest in plant genetics pervaded my education. As a graduate student, I learned how to breed crop cultivars. As a postdoc, I learned how to apply DNA tools to accelerate breeding of crop cultivars. And my lab has continued in these activities, developing and implementing tools for crop improvement in collaboration with breeders of the crops that provide us with our briefs, our currency, our vegetables and oils, our grains, our fibers, and other medicinal products that are essential to life. In the course of this work, we built up quite a lot of DNA information, eventually building up enough that we could begin to literally connect the dots between the genomes of different organisms, such as our major crops and botanical models, and even begin to make inferences about the nature of the common ancestor of all crops that somehow managed to evade being eaten by a dinosaur 150 million years ago. Through the course of this work, we found again and again the importance of a phenomenon called polyploidy, which is a genetic accident that results in a genome being multiplied twice or 3 times or 4 times or sometimes even 5 or 6 times or more, literally overnight. Polyploidy is very common in angiosperms, more common than in any other taxon known, even to the point of the crowning glory, which is canola or Brassica napus, that's multiplied its genome 72 times relative to the genome of the common ancestor of all angiosperms. Our ongoing work continues to focus on trying to fill in the gaps in this picture of angiosperm evolution, connecting more dots from more genomes, while also continuing to contribute to conventional crop improvement. A particular interest is in studying very recently formed polyploids, where we hope to be able to see literally in real time the processes and events that have given rise to botanical diversity, enabling the angiosperms to become the planet's dominant vegetation and also to provide essential ecosystem services. Thank you.
Great, thank you. Oh, that was great. Always good to learn a little bit about history and get back to our origins and roots, right guys? Speaking of roots, our next contestant is a guy who is so far underground you might think he was a spy. Although, come to think of it, he is a reputed member of the so-called Sunflower Mafia. I'm talking about Alan Boucher from the Donovan Lab, and he'll be talking to us today about the roots of plant adaptation. There you go.
The future of crop production, the future of food production is a huge concern, particularly in third world underdeveloped countries that are expected to have the greatest population increases. Ironically, it's these same countries that typically have the lowest fertility soil, the highest susceptibility to drought, and yet the least access to fertilizers. Luckily in the United States, fertilizer availability is not really considered a current issue, although our main source of phosphorus fertilizers, phosphate rock, is expected to be entirely depleted within our lifetimes. Clearly, in order to keep pace with growing populations, we're going to need to develop crops that can better tolerate low nutrient, low water, droughted conditions. Trouble is, the majority of our staple food crops today have been bred under high resource conditions for decades. And as a result, most of our food crops exhibit a sharp decline in yield in the absence of high fertilizer inputs. In order to develop crops that can better tolerate these low nutrient dry conditions, we need to instead look to the wild relatives of these crops that can already adapt to those conditions. For example, we can look at the cultivated sunflower up here on the top left, which is grown in about 2 million acres in the United States alone. It's related to wild species such as this one here on the left, which is native to the drought-prone, salty habitats of the Atlantic coast, or this species in the center here, which grows in the low-nutrient desert soils of the Great Basin Desert. The goal of my dissertation research is to understand how these wild sunflower species are able to acquire the water and the nutrients that they need in these extreme environments. In order to understand nutrient and water acquisition, we need to look at the roots, and that's what I do for my research. I study three main aspects of the root systems of these wild sunflower species. First, I study the fine roots, the thinnest portions of those root systems. The reason that's important is that the fine roots encompass the vast majority of nutrient uptake in plant root systems. I measure their size, shape, and production. Second, I study root system architecture, the actual three-dimensional deployment of roots in the soil. After all, nutrient uptake depends not only on the size and the shape of the fine roots, but also where the plant physically places them. Third and finally, all plant roots secrete various chemical compounds into the soil. Certain compounds secreted by plants have been shown to chemically interact with soil constituents, releasing nutrients that were previously unavailable to the plant. I study the composition and the specific nature of these compounds secreted by wild sunflower roots. It's my hope that studying these three main groups of traits, fine roots, root system architecture, and root secretions, that we can better understand how these wild sunflower species are adapted to those low nutrient, tough environments. Plant breeders can then take this information and be better informed on how to develop sunflower cultivars that can be grown with less fertilizer requirement and less irrigation requirement. Hopefully, research in sunflower and the related and relatives of related crops can help us achieve this goal within our lifetimes. Thank you.
Can I just say, as an objective audience member, I think these are some beautiful plants. All right. For our next contestant from Team Faculty, we've got Dr. Xiaoyu Zhang, and he's going to help us take a closer look at chromatin modification in plant stem cells. Do you want me to?
Yes, please. Just remember, you see the tallest tree today on my slide. So this is a tree that Alex sent me, and there are a few dots here that are actually, you can probably see organisms that we normally refer to as humans. So at the time of discovery, this was the tallest tree in the world and was about 370 feet tall. And since it was discovered, it grew a couple of feet taller still. It's probably around a couple thousand years old, and it's nowhere near the oldest tree discovered on Earth, which is 9,550 years old. Okay. So we don't work on trees exactly. We work on our Rhabdopsis. It's very similar to trees. The reason I'm telling you this is because really the whole above ground part of these plants are produced by a very small group of cells called the stem cells in the shoot apical meristem called the in the center of the shoot apical meristem. It's going to do two things. They keep on dividing and keep a small population as stem cells. So this is a maintenance part. And they also supply cells to other zones to differentiate into all the above ground tissues that you see. So based on gene expression differences, you can define a few different zones that are going to do very different things as the plant develops. Okay? So we work on chromatin modification. We came to realize that the modifications of chromatin probably plays a pretty important role in regulating the balance between this maintenance and the differentiation part. And the reason is, for example, this is a mutant that cannot trimethylate lysine 27 on histone H3. Okay, so what you're seeing here is actually a double mutant plant. Okay? The really good thing about this plant is it's immortal. So basically it lives forever. Okay? The bad thing is it's going to be looking like this forever. So it's a ball of undifferentiated stem cells. So the problem with this mutant is it cannot differentiate. Okay, so this is all stem cells cannot differentiate. Now this is another mutant that cannot modify another residue on histone H4. The problem with this mutant is, so normally the apical meristome is keep on dividing and providing cells to produce more flowers in this case. But this mutant is going to produce a few flowers and then quit and ends in the terminal flower. Okay? So developmentally speaking, the problem with that mutant is it cannot maintain the stem cell population. Differentiation, no problem. Maintenance, big problem. Okay? So what we really want to do is to figure out what happens in those tiny little cells. We need a few 100,000 of those. Each plant we can get a few dozen. So we are developing a high throughput tagging mechanism that we can use affinity purification, so bypass all the tedious work. You just have to grind up the plant and then do experiment, and you can figure out what's happening in the cells as they're being maintained and differentiated into lateral organs. Okay? So this is being done in corn, so we're looking at specifically these cells to figure out how they're maintained, how they transition from one state to another. And hopefully in a year or two, when the corns are old enough or big enough, we can do some experiments.
Not gonna do that in front of a room full of people. Whew. Xiaoyu, when you said you were gonna show us the world's tallest plant, I started having panic attacks about like some Godzilla Arabidopsis or something. Whew. All right. Up next, we have our last graduate student presenter, Uma the Tornado Nagenja from the Peterson Lab, and her talk is titled Disturbed Relationships. Can tornadoes change plant-soil interactions?
In August 2005, Hurricane Katrina blew through my hometown. I came back to find houses moved off their foundations, magnificent trees uprooted or snapped in two. Natural disturbances like these are very powerful, yes. But to me, what's even more powerful is what happens afterwards. Recovery and regrowth. We saw this in New Orleans, that people can get together and rebuild. But I'll tell you what, forests are even better at it. They do it all the time. We see landscapes that look barren one year. The next year, they're full of lush green life. When I saw this after Katrina, I wanted to know more. And it turns out we know a good deal about what species come back, but we haven't looked as much at how tornadoes change, how species interact. And species interactions are really important for ecosystems. Take plants and soil, for instance. In 1 gram of soil, you have millions of bacteria and fungi doing really important jobs. Soil is how plants get the resources they need. And they can't just get up and walk to a new location that has better stuff. So they're at the mercy of those soil organisms to either help them or take advantage of them. It's the relationship between the plants and soil that can determine whether that tree can spread out and dominate an ecosystem or have a diversity of neighbors. And it's not as simple as friend or enemy. It's also about how close that relationship is. You have some friends that you're really close with, like your best friends, but also people you just share cat pictures with on Facebook. The same goes for negative interactions. There's arch enemies like the Joker who just wants to get at Batman. Also some guys who are just jerks to everybody. The specialist relationships are like the best friends of the arch enemies. They really just involve a few species. There are others that are generalists that affect just about everybody. And because they affect just about everybody, it's the specialists that can make it or break it for a particular plant. What I expect to see after a tornado, such an abrupt change in the environment, is that the generalists would be more able to survive a disaster, which means you'd end up with an ecosystem that's full of Facebook friends and jerks, which affect everybody equally. So any disadvantage or advantage that a tree would have in an ordinary situation wouldn't necessarily be there. And although I first got interested in this from Hurricane Katrina, this is an issue for not just the Gulf Coast. The past few years, we've seen Hurricane Sandy and tornadoes ripping through Oklahoma and Alabama and Georgia. We're seeing stronger hurricanes and more frequent tornadoes, especially in the eastern U.S. So I want to see how these changes to our environment could affect our forests here in Georgia.
Wow, I don't know about you guys, but that presentation just blew me away. And that concludes all of our graduate student presentations for today. And last but not least, we've got our final from team faculty, Dr. Mark Farmer, and his talk is entitled Moving Towards Answering Life's Biggest Question. And right button when you're ready.
In my opinion, there's two really big questions in biology. The first is the origin of life itself. How did life emerge from non-living biochemistry? The second question is, from those earliest cells, how did it diversify into the wondrous, beautiful world that we see today of biodiversity? And that's a question that was largely answered by Charles Darwin. My research focuses on sort of the intersection between those two. Namely, how did the complex cells that give rise to all the plants, animals, and fungi first arise? And in order to do this, I study a group of organisms, single-celled organisms, that still retain some of those, I think, original features. So when I think about biology, I don't think just about how life is today, but I try to imagine how it was 1, even 2 billion years ago. In my laboratory, we're studying one such structure, the cilium. And if you know anything about cilia, you may have heard them referred to as little tail-like structures on cells that kind of push a cell through the water. But in the organism that I work with, Paranema, it uses its cilium in a variety of different ways. Sure, it can use it to move through the substrate, but it can also use it to feed itself. It can use it to detect obstacles in its way. It can use it to sense light. It can even use it for lovemaking. And I'm not going to show that one. We think that the mechanism by which this takes place is related to a group of proteins known as IFT that are within the cilium. And the IFT complex actually moves up one side of railroad tracks, deposits its cargo at the growing end, and comes back down the other side. And in doing so, it actually changes the shape of the membrane of the cilium. And we think that this small change in shape is what's responsible for allowing these cells to do something unusual, which is to glide along the substrate. So what Paranema does is it lays its flagellum down in front, its cilium in front of it, and basically glides along in what I think is a very, very ancient form of cell motility. Now, all complex organisms, including ginkgos and geckos, still have these IFT complexes. You and I still have them, and I think that they still play a role in our biology, which is why understanding how Paranema moves does have relevance to our understanding of our own biology. Now, if someone came from another galaxy and wanted to know how humans construct buildings, we wouldn't hand them the blueprints to a skyscraper. We would show them a hut with its simple ideas of a wall and a roof and a door by which you go in and out. So in terms of what I do in biology, I study the huts. Thank you.
See, this time I was actually going to tell you guys to applaud, but you beat me to it. All right, so that concludes all of our 3-minute throwdown talks for this year. So now what we're going to do after, get everyone all loosened up is to take about a 10-minute break, and this will give the judges time to finish their tallies and for you guys to turn in your People's Choice ballots, and we can tally those up and award some winners. So refreshments are outside still if you guys would like to, and we'll reconvene in a few. Okay, first I'd like to give a brief thank you to everybody who helped make this possible, namely Plant Biology, the Grad Student Association, and the whole department as well. That's sort of where the idea originated to make this sort of a home event, and I think it turned out really well. Would you guys agree? Woo! Also, a big thanks to Beth Richardson in the back, who's been helping record this so it'll exist for all perpetuity, which I'm sure you guys are really excited about. And for folks who helped out making this possible, Megan was really helpful in getting the word out and sending flyers everywhere. Carlina was awesome with her timer of doom. David made that awesome poster that got sent out everywhere and confused people about the SmackDown, and that was great. And Elise has made our wonderful trophy here, which is what you guys are all competing for, the fame and the glory and the honor. Also, you guys who participated, you did a phenomenal job, and I'm sure it was stressful out the wazoo, but you guys did great. And finally, a big thanks to our judges who helped make sure that everything was staying on track. And we've got some cool plant biology swag for you guys. I think we've got 2 drinking vessels, I think, which were our departmental fundraisers. We like to drink a lot, I guess. And last but not least, her name is not on here because she was the one who made the PowerPoint and, of course, wouldn't put herself on here. But I'd like to give a huge thanks to Jess Stevens, who made this entire thing happen. She has done a tremendous amount of work making this a huge success, and I don't know if whoever comes after you as our PBGSA peer evaluator will make this happen in the future, but thank you so much, Jess. And thanks
Caitlin for being an awesome MC.
All right, okay. So now the part you guys are really excited to hear about are the awards. So first, we're going to announce the best grad student presentation. And where are my results? Oh, here we go. Okay. With a total of 168 points out of a possible, they got a lot of points. Please give a huge shout out to Miss Uma Nagendra. Oh, and she won a, what is it, Jess?
$20 gift certificate to Cali and Tito's.
Woo! All right, next up is the best faculty presentation. And with a total of 154 points, we have Dr. Shumei Chang. Oh, okay. And
$20 for that one, too.
For Cali and Tito's. All right, next is the People's Choice Award, and the award for that is a totally blinged out koozie, which can be featured at our Final Friday events, so you can brag to everyone that that was
starting
you.
up next month again?
Yes, and the winner of that swag is Dr. Michelle Momini. And finally, the moment you guys have all been waiting for is the end all, be all, who was better, faculty or grad students, and the winner with, it was a total of 869 to 825 points. So it's kind of close, guys, but the winner are the graduate students! Sure. All right. I think that's all we have for you guys. So thank you so much again. This has been a blast.